let's continue our section with next speaker, Dr. Francesca Briganti. Dr. Briganti is a molecular biologist interested in understanding the molecular causes of human disease in using this knowledge to design targeted therapy strategies. She is an instructor at McCullough Lab at Stanford. Her scientific interests include cardiac physiology, disease mechanism, and drug development. Dr. Briganti received her PhD jointly from EMBL and the University of Heidelberg. During her PhD, she studied new targeted therapy approach for the dilated cardiomyopathy, and she identified a novel potential therapeutic strategy that has been published and patented. Please welcome Dr. Briganti.
Hi, thank you so much for having me as a speaker and for such a kind introduction. Today I'll tell you a little bit about my research, which focuses on bridging the gap between genetic mutation, molecular mechanisms of disease, and personalized therapeutics. As you've heard, I am a molecular biologist by training, and I've always been interested in molecular mechanisms of disease. But first of all, I want to ask the question, why do we need personalized therapeutics for cardiac disease? And here is a graph. I'm sure all of you are familiar with this kind of data, but it really helps me putting them on a graph. So these are the 3 top leading causes of death in the United States. This data has been derived from the CDC. And heart disease is the first cause of death in the United States, adjusted by age. I have also derived the following data from the FDA for drugs that have approved in 2023. And you can see there is a huge disproportion between the number of drugs approved for cancer and the number of drugs approved for cardiac disease. In particular, there's only been 1 drug approved for heart disease in 2023. On the contrary, there have been 14 drugs approved for cancer. And of course, cancer is an umbrella term. However, what I find very interesting that many of these drugs are personalized to a certain extent. They are mutation positive, mutation negative, biomarker positive, biomarker negative. However, only 1 drug was approved for cardiovascular disease in 2023. This is SGLT inhibitors for heart failure, and we don't really understand the mechanism for this drug. I think that of all heart disease, dilated cardiomyopathy presents the perfect opportunity for the development of personalized therapeutics. Why is that? Some of the data on this slide, again, you're very familiar with. Dilated cardiomyopathy is a leading cause of heart failure, affecting about 1.2 million Americans each year. It has variable clinical presentation, and what, as a molecular biologist, is interesting to me is that this is paralleled by variable etiology. Even when you look only at genetic mutations, these mutations, differently from other diseases, don't fall into the same category, nor where you look at molecular mechanism, nor where you look at subcellular localization. Here you can see my 3 favorite dilated cardiomyopathy genes, PLN, RBM20, and troponin T. Obviously, you're very familiar, but they not only have different molecular function, calcium regulator, splicing regulator, sarcomeric proteins, but they also localize in 3 different cellular compartment. What I believe is that the difference in the molecular mechanisms of dilated cardiomyopathy can be addressed in the development of new therapeutics. Another very interesting fact about dilated cardiomyopathy is that, of course, there are many causes, and about half of DCM cases are familiar, so they have a genetic component, but as much as a third is monogenic, which means we can relatively easily model this in vitro. And I will go through some data I have previously published just to exemplify what I mean by modeling it in vitro. I'll be very quick because this is published. But back in 2016, actually, we identified a patient from a family in which dilated cardiomyopathy was segregating. We did some genetic analysis, and we found that this patient and other affected individual from his family had a new variant in RBM20. We were able to introduce this mutation in a healthy donor cells, iPS-derived iPS, and then differentiate these cells into cardiomyocytes. These cells are beautiful. As many of you are familiar with them, they beat spontaneously in vitro. And then I started collaborating with Mark Mercola here at Stanford to quantify contractile function of RBM20 wild-type and RBM20 mutant iPS-derived cardiomyocytes. And we were able to show that there was a phenotype in the patients compared to the control and in our knock-in lines. Of course, I was not the first one, nor the only one to show that DCM mutation caused a phenotype in iPS-derived cardiomyocytes. These are two other works from Stanford. In particular, you can see here that a patient iPS-derived cardiomyocytes compared to their isogenic control have a contractility defect with mutation in PLN, and similarly with mutations in troponin T. The reason I'm showing this is because all following study will be using these lines that have already been published, so we already know they have a phenotype in vitro. From here, of course, to answer the bigger question of can we identify mechanism-specific compensatory pathways, therapeutic targets for dilated cardiomyopathy, how do we go about that? And I worked really hard during my postdoc to develop an unbiased screen that will allow me to do that. And I'll walk you a little bit through those steps. First of all, just to summarize here, my goal is to gain comprehensive mechanistic insight into etiology-specific DCM compensation through the analysis of microRNA and their targets, and you will see why in just a minute. Here is my strategy. I tested the effect of 66 microRNA that had been previously shown to be upregulated in end-stage heart failure in iPS-derived cardiomyocytes from patients with mutations in these three genes. And then I compared the effect of those microRNA on the three different lines and some isogenic controls. And once again, here you will see a schematic of how the screen was set up. We have three patients, one with each of the three mutations I've shown before. Those lines have been previously published. They are the same as the papers. Ioannis had already introduced a double mutation in the troponin T patients, as well as corrected the mutation to obtain an isogenic healthy control. All of these lines were differentiated into iPS-derived cardiomyocytes. They were mature for 2 months, according to a protocol that has been earlier described by Anna this morning. 2-month-old iPS-derived cardiomyocytes were replated into 384 well plates. The cells were allowed 4 days for recoveries. All cells were beating very consistently on the 2nd days after replating. That's actually a checkpoint, and they have to pass it to proceed through the experiment. On the 4th day, each well was transfected with an individual microRNA or a control. This experiment was done with 8 technical replicates, means for each experiment, 8 wells were transfected with each microRNA or SI. And it was repeated twice for each cell line. Then the cells were allowed 8 days to recover and analyzed again for contractility with our kinetic imaging cytometer, which again was very kindly described this morning by Anna. And of course, we can measure different parameters. The ones I'll be focusing on today are mostly contractile rate, which is the maximum slope of the contraction part of the curve, and the D peak, also known as contraction amplitude, which is the maximum displacement. And as you can see from panel C, of course, different microRNA had different effect on those different parameters. Here from bottom, you have wild-type cells, and then you have all the iPS, all the mutant cells. And then you can see in orange is the contractile rate, and in green is the contractile amplitude, contractile rate, contractile amplitude, and so on and so forth. So first of all, you might notice that it's a lot easier to improve contractile function in wild-type cells than it is in dilated cardiomyopathy cells. That was surprising. You will also see that very often if you have blue, so if you improve contractile function in DCM, you'll almost always also improve it in wild-type cells. Because these are immature cardiomyocytes, as much as we want to mature them, of course, they're still immature, and we wanted to avoid any problem associated with improved maturation of the cardiomyocytes. We really wanted to focus on what was therapeutic. We selected 2 microRNA that showed an effect in disease cells, but not in wild-type cells. And here you can see the name of these two microRNA, miR-181 and miR-452. You can see miR-181 improved contractile function in all DCM lines, but not in the corrected line. Contractile amplitude is on top and contractile rate is at the bottom. miR-452 was particularly interesting because it did not improve contractile amplitude in RBM20 or PLN, but it significantly improved contractile amplitude in the troponin T cells. And it actually worked both in the patient-derived cells and the homozygote mutant cell. This, you might remember, is the same patient, just with the correction. And to me, that was very reassuring that the exact same genetic background, with the only exception of the mutation, did not respond to miR-452. Similar data is shown for contractile rate at the bottom. However, that is still one patient, and we want to make sure we're not going after something funny that a single person's genome is doing. And so I selected 3 more iPS-derived cardiomyocyte line with the same mutation in troponin T, and I was able to show that miR-452 was still able to improve contractility in those lines, kind of corroborating the data I've shown so far. Of course, the first question you might ask, and a little bit because I'm a molecular biologist, like, well, what do these microRNAs do? Let's look at how they change the transcriptome. And sure enough, you can see that there are genes that are upregulated by miR-181 and genes that are upregulated by miR-452 and genes that are downregulated by either or the other. But something that really caught my attention was the principal component analysis. On the x-axis, you can see principal component 1. This explains about 40% of the variability, and it very clearly separates miR-181 from miR-452. However, what's interesting is on principal component 2, this still explains 25%, about a quarter of the variability, and I'm clearly separating the controls from all of the treatments. And sure enough, this is also visible in the heat map here. There are genes that are downregulated by both microRNA and genes that are downregulated by both microRNAs. And when I look at what pathways are affected by both microRNA, there are some that are, not really surprising. Response to hypoxia, metabolism comes up a lot. But then the question was, are these gene changes causing the cardiomyocytes to beat better? Or are they a consequence of the fact that the cardiomyocytes are beating better? Is that a causal gene or is it a biomarker? They're both interesting, but when you want to go after therapeutic targets, you need to get to the root cause. How do we go about that? Well, I took a biochemistry approach, and I went after mRNAs that were directly bound by the microRNAs in question. Here is just a little summary. The microRNAs are transfected in iPS-derived cardiomyocytes with the troponin T mutations. Then the cells are cross-linked, lysed, and then the AGO-IP complex is immunoprecipitated, the RNA is fragmented, and then this intramolecular chimeric molecule ligation reaction is performed so that we form a chimeric molecule. You can see this is then used for library preparation, and when you read your library, what you will see is the first 22 nucleotides is the microRNA, and the next how many nucleotides will tell you what's the targeted mRNA. What did we find here? Well, we found that many genes are bound by either miR-181 or miR-452. That was about 600 to 700, which is a little bit too many to follow up. We decided to overlap this with genes that we knew were downregulated in the RNA-seq. And is this approach giving us real targets? What I decided to do was to extract the sequences that had been that represent this 66 and this 104 mRNA here, and look at motifs. This is produced with MEME, and you can see I rev-comped the sequence for you just to make it easier to follow, but I was very happy to see that 100% of the sequences bound and downregulated by miR-181 had a motif that looked very much like miR-181 seed sequence. And for miR-452, the percent was a little lower, but I thought it was still pretty good. And I'm happy to discuss why I think this is happening offline. Now from here, I selected these 66 and this 104. I also added 30 controls. These were genes that were bound by the microRNA but not downregulated, 15 from miR-181 and 15 from miR-452. And I went through my screening pipeline again. This means I have 2 months old iPS-derived cardiomyocytes, replate them in 384 well plates. I do quality checks, make sure they start beating on by the second day. Then I transfect individually 200 size connection in 384 well plate formats, and then analyze them for contractility. And I've done this in 3 iPS-derived cardiomyocytes line with technical and biological replicates. And here you can see that over 100 genes in this screen showed an improvement in contractile amplitude and rate, and some also in contract and relaxation rate. And this was true for both miR-452 and miR-181 target. One slightly disappointing point of this slide is that filtering for downregulation didn't really improve the proportion of genes that had a positive effect. Obviously, I believe this has to do with the mechanism of action of microRNA, which lead to suppression of protein expression, not necessarily by leading to mRNA degradation, especially at the time this experiment was performed. And obviously, that does show that there is room for improving this pipeline. However, I do think that 100 gene is a good starting point for starting to look for therapeutic targets. When we look a little bit closer, we find that of the 100 genes on this slide that show some blue, 5 have been identified as cardioprotective in animal models. And at least one of these genes has progressed through clinical trial. This clinical trial specifically was halted due to off-target toxicity of the small molecule that was being used. But I think overall, this indicates that we are on the right path for identifying therapeutic targets. Also, I do want to give you a little bit more data here. This is obviously unpublished. Hopefully will be published soon. But some of the things we've started doing is, once again, 100 genes is a little bit too many to go after, but we can start assembling them in pathway and see if there's a specific pathway that can be targeted. And we think we've identified at least one. One of our favorite genes is BAC1. This is a transcription factor. It suppresses hypoxia response genes. And then another gene we think belongs to the same pathway is PRKG2. So PRKG2 is a negative regulator of MPK, so its suppression leads to activation of MPK, which leads to activation of NRF1 and 2, which has an opposite effect of BAC1 on the same target gene. So we think this is what's happening, and we're currently working on this. Some other reasons I think this pathway is interesting. BAC1 knockout mouse exists. It has absolutely no phenotype that the researchers have been able to uncover at baseline. However, these mice are protected from pressure overload and ischemia reperfusion. MPK activation, I'm sure many of you are familiar. There's been plenty of studies showing that this can be targeted therapeutics. It's being investigated by multiple groups. PRKG2 is very controversial. We don't really understand why we think it's because it's a negative regulator of MPK, and so it eventually leads to MPK activation. However, mutation in PRKG2 have been shown to cause dilated cardiomyopathy. When we looked a little bit closer at this mutation, it showed that those mutations were actually a gain of function, so maybe we can make sense of this information, but still it is a little bit more controversial, and it was worth mentioning. This brings me to the end of my talk. Here is a summary. So we have identified beneficial microRNAs, and we have identified microRNA targets, and we have tested these targets and found that at least 100 of them improved contractile function in vitro in iPS-derived cardiomyocytes with mutation in troponin T. And here is a final summary of the pipeline I have been developing during the years of using a primary screen, using microRNA to test the effect of not one gene at the time, but multiple genes at the time. We estimated about 8,000 genes were probed in the primary screen. And then through target identification, we come down to about 200 genes. And then what we were hoping when we started this project was to go from here to here. But then we found ourselves stuck in about 100 genes that had a positive effect in the secondary screen. I think that's really exciting, and I look forward to understanding that better. These are some of the things we're doing. You got a small glimpse of building gene network. We're also working with a group in San Diego to do system biology analysis of these genes, integrate human data through GWAS studies, and then include additional disease models, both in vitro and in vivo. The idea is to come down to about 2 or 5 genes that are worth testing in preclinical settings. Hopefully so far, with my talk today, I have convinced you that it is possible to identify new novel therapeutic targets for dilated cardiomyopathy by combining iPS-derived cardiomyocytes model of DCM and high-throughput functional genomic screening technology. And here is my acknowledgement slide. This work was, of course, done in Professor Mercola's lab at Stanford. And here are people that through the years helped at different stages. Here is a list of trainees that helped with the project. And then I have other collaborators in other groups at Stanford, as well as other University of Cincinnati, the University of San Diego, my PhD institution in Germany, and some more collaborators in Berlin. Here are also my funding sources, the Cure Plan. I was recently awarded a K99. The Cardiovascular Institute has been very supportive as well. I'll be starting my own lab in the summer at UC San Diego, so that's next. And if anyone is interested in this kind of research and beautiful weather, come talk to me, obviously. And I'll take any question. Thank you.
Thanks, Dr. Briganti. Congratulations.
Thank you.
Any questions?
Francesca, great talk. You said in the beginning that your original screen is 66 microRNAs that upregulate in heart failure, and you have 100 targets. So theoretically, those targets should be also downregulated in heart failure. Have you, actually 2 questions, have you looked at the expression of those genes in heart failure, and whether these are good targets if they are already downregulated?
Yes, that's a great question. And yes, I've done some of those things. And the answer is that some of those genes are upregulated and some of those genes are downregulated in heart failure. Some of the things to keep in mind is that the microRNA are overexpressed at non-physiological levels. So they're expressed a lot more than they will be in a real heart. And the targets we're after are not really the targets that you will find in heart failure, but are those that show a therapeutic effect. So I'm going to say it doesn't matter what they do in heart failure, but if inhibiting them improves contractile function, that is what we're after. Some of the targets are upregulated in troponin T mutant iPS-derived cardiomyocytes. And we think that the microRNA is having a normalizing effect on those specific genes.
Next question.
Great talk. So I wanted to ask something follow up to the same question. You specifically mentioned end-stage heart failure. So I know you probably know this, but I wanted you to address a little bit more. Is there any way that we can look at some targets in the progression before the end stage? Are there any lists like that? Thank you.
Yes, thank you so much for that question. So once again, to keep in mind that the microRNA are a tool as an entry point to identifying targets. So we're not claiming that is what's going on in end-stage heart failure. We just needed a manageable set of tests to run, and 66 was a good number. Another nice thing about microRNA is that with the only exception of LET7, there is no known mechanism for degrading microRNA. So unless the cells are dividing, those microRNAs stick around. And cardiomyocytes are notoriously non-dividing cells. So as a molecular biologist, I like to believe that looking at microRNA has a deeper value than looking at mRNAs, also in end-stage heart failure samples.
Oh, hi, nice talk. I think you mentioned in the end that you want to target the NRF2 antioxidant kind of pathway. So I wonder whether have you noticed there's a high baseline oxidative stress in this troponin 2 DCM line, and maybe this underlies some contractile dysfunction. And if you ever, modulate this oxidative stress, do you see still improving the functional change?
Yes, thank you so much for that question. That experiment is being finalized, so it has been done, and I don't have the data to share today. Thank you.
I have a quick question. The microRNA that you identified, are they specifically expressed in cardiomyocyte, or they are also expressed in other cell type in the heart failure?
The microRNAs
yes.
that were tested originally, no, many of them are ubiquitous.
Thank you. Any questions?
Hi, wonderful talk. Quick question. So MIR452, it's X-linked, right? It's expressed in the X chromosome. Do you see any gender differences?
I have not looked.
You never looked at that? Okay.
Thanks. Thank you.